The human brain is the most important organ of the human body. Over 250,000 people died of brain cancer around the world in 2023. In order to cure the brain cancer, we come up with a drug delivery method using DNA origami to deliver the drug into the brain and cure the cancer. However, the human brain contains a barrier to block other materials from entering the brain. This barrier is called the blood-brain barrier, aka BBB. This important barrier consists of four parts: parasite cells, basement membrane, endothelial cells, and tight junctions. If there is a protein in the blood vein of the brain, the protein will be blocked from entering the brain. But if the protein binds with the transferrin aptamer, which is a small strand of a DNA, the protein can pass through the blood-brain barrier by transcytosis. In our study, we use a DNA material called DNA origami as the carrier of the drug. We designed the transferrin aptamer on the side of the DNA origami to let the carrier pass through the blood-brain barrier. This way, the drug can be delivered into the brain and cure the cancer. First, we need a long and circular single-stranded DNA, which is called the DNA scaffold. Next, the DNA scaffold is mixed with several short, single-stranded DNA called DNA staples, which are specifically designed to bind to the particular position on the DNA scaffold. After being treated with a certain thermal cycling, the staples bind to the scaffold, forming a particular shape. The staple nucleotides pair with the scaffold in the corresponding position. By properly designed, the DNA can be formed into the required shape. We use the DNA scaffold M13MP18 P7249, which contains 7,249 nucleotides. And then we designed a series of DNA staples and mixed with the scaffold. Allows the DNA scaffold to form a triangle which is called the Rothemund triangle. We replace the staples on the middle of the side of the triangle to the staple with transferrin aptamer. This is our drug carrier material. If we see the shape of the scaffold only, this is what it looks like. First, we designed a microfluidic device made of PDMS, and then we loaded the blood-brain cell including parasite cells, basement membranes, and endothelial cells on the microfluidic device. After that, we add the origami solution with a transferrin aptamer into the device. The origami should pass through the blood-brain barrier cells. If we further add the fluorophore on the DNA origami, we can observe the fluorescence on the other side of the microfluidic device.